Hello everyone, we're going to be doing a full course review of the content for the EOC end of course biology exam in North Carolina. Now if you're watching from another state, this video still might be helpful for you, but you always want to check on your state standards and check with your teacher to make sure you're studying the right things for your exams. Now this video may not cover the content in the order that you studied, and it is not everything that you learned in biology this year, but hopefully it will help you with your review, give you some refreshers on old ideas and ideas of things that you still need to study in preparation for your exam. So grab your notes, get ready, and let's start the review. All right, let's start with the 4 major macromolecules that are necessary for life. You might have also heard these called or referred to as biomolecules or organic compounds, but the big 4 categories are nucleic acids, proteins, carbohydrates, and lipids. Nucleic acids are going to store and transmit our genetic information in the form of RNA and DNA. Proteins are going to do all the jobs for the cells, so they're responsible for structure in our muscles, in our hair, in our nails. They can send signals, they can form antibodies, and they're even enzymes, which can speed up or facilitate chemical reactions. Carbohydrates' main function is to provide energy, quick energy in the cell. We can also find carbohydrates in cell walls in certain organisms. And then, of course, lipids. Here we have an example of a phospholipid, which is going to be a major component of the cell membrane and surround all cells, but lipids are also used for long-term energy, and they can be signaling molecules as well. The monomer, or the basic structure of each organic compound, are shown here. So we've got nucleic acids are made of nucleotides. This is one nucleotide in the picture. Proteins are made of amino acids. We have several amino acids linked up to form a polypeptide chain here. Carbohydrates, monomers, are monosaccharides, and lipid monomers are typically things like fatty acids and glycerols, which form things like triglycerides, but lipids are pretty diverse as well. Now, each of these molecules are made of some basic elements. I have these here. It's important to know that the most important elements to living things are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Now, that's kind of a long list, so a good way to remember it is CHNOPS for carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And these elements get into living things through a multitude of different cycles, like the carbon cycle or the nitrogen cycle, which we'll review a little bit later on in this video. A good way to remember all 4 categories of organic compounds or biological macromolecules is clean later, party now for carbohydrates, lipids, proteins, and nucleic acids. Now, I like to add especially now to this mnemonic, clean later, party especially now, because that reminds us that enzymes are proteins, which are a major and important category of protein. All right, so let's talk about why enzymes are important. Remember, these are proteins, and they're going to speed up chemical reactions, so we call them catalysts. Some examples of enzymes that you might see are ligase, helicase, polymerase, amylase, and you don't need to memorize all of these, but notice that each of the enzymes here ends in the letters ASE, which is a very common word part indicator to tell us that a molecule is an enzyme. So if you see a molecule that ends in ASE, it's probably an enzyme. Enzymes are important in medicine and health, the environment, ecology, but most importantly, enzymes are reusable and specific to their substrate. So this is an enzyme, and the molecule it acts on is a substrate. Enzymes can be reused after each reaction. So as we see here, a substrate would interact with an enzyme, conformational change, and turn into the products, but the enzyme is unchanged at the end of that reaction. Again, don't forget that enzymes are proteins, so that means that they are made of amino acids just like any other protein, and they can be affected by certain environmental factors like temperature or pH, and they may have an optimum pH or temperature they work best. But if the temperature gets too high, for example, in this enzyme, if we see temperatures that are too high, the bonds between the amino acids can be disrupted, and this enzyme can denature or unfold, and then it will not act on its substrate. And so we might see a graph like this here on a test where we're asked what the optimum pH is for a particular enzyme. And so we're going to look here on the graph to where the point is the highest, and we can see that it goes down to about 8. So a pH of 8 means that this enzyme will perform best in an environment that has that pH. Of course, enzymes are very important in cells, but there's a lot of other components and different functional parts of cells that we're going to review today. Remember that all cells and all living things are surrounded by a membrane, that's that phospholipid bilayer. They contain genetic information in the form of DNA, and they do have cytoplasm. But we do see differences between prokaryotic and eukaryotic cells. So prokaryotic cells are a lot simpler. They do have a membrane and ribosomes and genetic information, but you see that it's free-floating encoding in the cell in what's called a nucleoid. Eukaryotic cells are much larger and more complex, and they have membrane-bound organelles, so organelles that are surrounded by a membrane of their own, which means that their reactions are compartmentalized and they can be more efficient and do more things. All bacteria are prokaryotic, and things like animals and plants and fungi, those are eukaryotic organisms. Most importantly, eukaryotic organisms have a nucleus which surrounds their genetic information. Prokaryotic cells, once again, do not. All right, so let's talk about some of those major organelles within the cell. I'm going to tell you what the function is, and I want you to try to think about what the name of the organelle is and what it might look like on a cell diagram, and I'll tell you what the answer is right after I read the definition. So, a large fluid-filled sac that stores nutrients and helps maintain cell structure in plants specifically is a vacuole. Now, animal cells have vacuoles as well, but in plant cells they can be much larger because that water storage component is very important. A membrane-bound sac that contains enzymes that break down waste products and damaged organelles, that is a lysosome. Now, on a diagram you might just see this as a circle with a membrane around it, but remember that lysosomes are often referred to as the garbage disposal of the cell because they're responsible for breaking down waste. An organelle responsible for modifying and packaging proteins and other molecules for transport within the cell or export from the cell is the Golgi apparatus or the Golgi body. Now, you might see that illustrated as a series of several different membranes like this here on a cell model. A network of tubules and sacs that helps transport proteins and other molecules within the cell, that's the endoplasmic reticulum or ER. Now, we see the ER here. This is the SER or the smooth ER, and this is the RER or the rough ER. And you'll notice the difference between the RER and the SER is that the RER or the rough endoplasmic reticulum contains ribosomes. A small structure that produces proteins in cells are the ribosomes, which are often illustrated as little dots within the cell, which we saw here and again on the RER. And a rigid structure that surrounds the cell membrane of plants and some other organisms like bacteria is the cell wall, which we can see can exist on eukaryotic organisms and on some prokaryotic organisms as well. Animal cells do not contain a cell wall. The gel-like substance inside the cell membrane is the cytoplasm, and an organelle in plant cells that uses sunlight to produce food is the chloroplast, which is often illustrated as a circle with these different stack-like structures, and we'll talk a little bit more about the chloroplast later when we get to photosynthesis. One more for today. The site of energy production within eukaryotic cells is a mitochondrion, or plural, the mitochondria. Remember, you probably heard that mitochondria are the powerhouse of the cell, and they're often illustrated as ovals with little squiggles. Those are the inner membrane folds of the mitochondria. Let's take a look one more time at this phospholipid bilayer. We'll talk about how molecules get in and out of the cell in a little bit, but remember that this is a type of lipid, a phospholipid. It's got a phosphate head and then a lipid tail. Lipids are hydrophobic, so these tail portions are water avoiding, and this head portion is hydrophilic, meaning it does interact well with water. So the way the phospholipids arrange themselves around the cell is in this double layer where we have the heads facing out and on the inner side of the cell, and these tails are pointing hidden away from the water or the cytoplasm. Now, again, all living things contain DNA, and DNA is found in the nucleus of eukaryotic organisms or in the nucleoid of prokaryotic organisms. DNA's monomers, remember, are nucleotides, so that means they contain a phosphate, a sugar, and a base. The sugar is called deoxyribose, hence the name deoxyribonucleic acid. Now, we talk about how DNA is our genetic code and it makes us who we are. How do we get from a DNA code in the sequence of different nucleotide bases to the actual physical characteristics that we have? Well, it's all about the central dogma of biology, which is also called protein synthesis, which means the process by which DNA is transcribed and translated into proteins, or how proteins are made. So you probably remember that we start with DNA, it gets transcribed into an mRNA message, and then that message will help tell the ribosomes how to build the correct sequence of amino acids to make a protein. So let's take a look at a model of how this works. We start with an original DNA strand in the nucleus. That DNA separates with the help of enzymes, and then another enzyme comes in and helps add RNA bases that are complementary to the DNA strand. And this forms an mRNA strand, which can leave the nucleus. And then that mRNA will go to the ribosome, where each set of 3 mRNA bases, so here GAG for example, will meet up with a tRNA molecule, and that will bring over an amino acid. Then those sets of 3 amino acids or codons will continue to be read by the ribosome, linking up more and more amino acids until we have a full protein sequence. Now, most proteins are dozens, if not hundreds, of amino acids long. This is just a really big simplification here. But the order of amino acids is going to determine how it folds on itself and becomes a functional protein. So let's practice how to do this. Remember that in RNA, it's the same base pairing rules, except we don't have T, so A pairs with U instead of T. So if you want to pause and try to write out the RNA transcription here, you can. The correct RNA from this DNA sequence is shown here. In order to figure out which amino acids are going to match up with each set of 3 bases, we'll use what's called a codon chart, and it can look either like this or like this. So let's practice with the circular one first. So we have an mRNA sequence. If you want to pause the video and try to figure out which amino acids are going to match up with each of these codons, go ahead. The correct answer is tyrosine, glycine, and isoleucine. Now let's do it all from DNA. So start with the DNA, make it RNA, and then try to find the amino acids that correspond to the RNA sequence. If you want to pause the video, go ahead and do it now. The correct answer is shown here. Here's our RNA sequence, and here are the amino acids that come from that RNA sequence. All right, a quick reminder about DNA and chromosomes. Remember that DNA is condensed in the form of chromosomes in cell, but this is actually a bad representation of that. The DNA is folded over tiny little proteins in super, super long strands, so it looks more like this rather than this inside the cell. And you can kind of see another representation of that folding here around these proteins and the twisting and the curling to form the condensed chromosomes. Now, the DNA will condense into chromosomes in order to prepare for cell division. And just a little bit of vocabulary reminder, within the cell, when that DNA is not condensed, it's in the form of chromatin, which we see here. And then one single duplicated chromosome looks like this X or butterfly figure. And then the identical strands of DNA on either side are called sister chromatids. So this is a sister chromatid, and this is a sister chromatid. The whole thing is a chromosome, and this here is chromatin. So if you need to go back and review that, that could be helpful for you. Now, before cell division, the DNA will condense into chromosomes, like we see here. And that is part of the cell cycle. So the cell cycle has a lot of different checkpoints that allow the cell to perform the processes and different parts of its life cycle that it needs to do in order to survive and then later on divide in the M stage. So most of the cell cycle is what's called interphase, which includes G1, S, and G2. And then the M phase is where it's going to go through cell division and split into 2 new cells. Now remember, DNA has to replicate or duplicate itself, and that happens during the S phase of the cell cycle, so that when it goes through mitosis, we don't end up with half the amount of DNA in each daughter cell. We end up with an exact copy of the DNA in each daughter cell. And then finally, this final stage here, cytokinesis, is when the cell actually physically splits into 2 new daughter cells. In order for that DNA to replicate, it's kind of similar to protein synthesis, where the DNA will be split apart and then new nucleotides will be added to each strand. So we end up with 2 strands of DNA, one old and one brand new with new nucleotides. And we see here A pairs with T, G pairs with C, et cetera, et cetera. And these strands are identical. We call this semi-conservative DNA replication. All right, so when we're actually going through mitosis, you might have heard, we go through a bunch of different stages, which you may or may not have had to memorize as PMAT, prophase, metaphase, anaphase, telophase, and then of course cytokinesis. But very quickly, the chromosomes are condensing before mitosis occurs. Prophase, the chromosomes are preparing for cell division. The nuclear envelope is dividing. These spindle fibers are going to come and attach at the center of the chromosomes as they align in the cellular equator in metaphase. In anaphase, sister chromatids are pulled apart towards opposite ends of the cell. In telophase, the new nuclear envelope starts to form, the chromosomes decondense back into chromatin, and then in cytokinesis, we have the actual splitting of the membrane into 2 new cells. All right, now you also probably heard of meiosis as well. A good way to remember the difference is mitosis is just regular growth and copying, and cell division, it's happening all the time in our bodies. So think about the growth that's happening in your toe, mitosis. But to make a whole new organism, we actually need cell division to go through meiosis. So to make me a whole new person, you would do meiosis. Now remember, cell division is just going to happen normally all the time in our bodies in order to grow. But we know that not every cell in our body is exactly the same. And so cells can differentiate or end up activating different parts of their DNA or in order to look structurally very different. So every organism will have the same DNA in all their cells, but in each cell we may have different parts of that DNA expressed or turned on in order to generate the proteins it needs to look the way it will. So different parts of the DNA are activated and expressed in muscle cells, and different parts of the DNA are activated and expressed in things like neurons. The cell cycle is pretty important, and there's a lot of, like I said, checkpoints that the cell has to go through, but sometimes those checkpoints, which are regulated by proteins, can get messed up, and that's when we have issues like cancer. So if the cell continues to divide when it shouldn't, and and we have uncontrolled cell growth. That is when the cells are going to divide and divide and divide in your own body, and your own cells can produce things like tumors, which can make people very sick, which is the disease of cancer. Usually these issues are called by mutations in the cell's DNA, and so the DNA is going to produce these different proteins called cyclins, and when the cyclins are disrupted or they're not produced at the right amount, the cell can continue with uncontrolled cell growth and cancer. It's important to talk about feedback within cells and organisms and ecosystems that help keep living things alive. Feedback loops are going to help us maintain homeostasis, which is a balance of conditions inside the body that organisms need to stay alive. So, for example, certain water levels, temperatures, heart rates. If organisms can't maintain homeostasis, it can often cause disease or death. So let's take a look at some of this feedback. For example, we have our blood pressure, which is regulated in the body. That's a type of feedback. So we might have a normal blood pressure to begin with. A stressor causes our blood pressure to rise above a normal point, and then receptors in our body are going to detect change. They're going to decrease our heart rate and increase the blood vessel diameter, so the size of our blood vessels, which will help our blood pressure return to normal. And we have that feedback mechanism working. Now, if this feedback wasn't working properly, we could have a continued high blood pressure rate, which could end up causing problems for the organism. Not just humans are going to experience feedback. These are what's called stomata, these little openings in plant leaves here. And these can open and close based on the amount of water or plant hormones or other stressors in the environment that are going to send signals for these guard cells, which are surrounding the stomata, to either close and prevent water from leaving the plant leaves or open up if they need to allow certain gases in, like carbon dioxide, in order for photosynthesis to occur. Now, cells can also regulate themselves by letting certain things in or out. And in general, we have three main types of cell transport that we'll talk about in biology: diffusion, facilitated diffusion, and active transport. Remember, diffusion is the movement of molecules from high to low, and so I like to use this slide metaphor to talk about it. If you think about a child going from high to low, it's like molecules going from a high concentration to a low concentration without any extra energy needed. Now, facilitated diffusion, you might think of a kid going down the slide with a little help, and often that's a molecule going from a high concentration to a low concentration with the help of a transport protein. An active transport, we would need to introduce more energy because that's the kid going from a low concentration to a high concentration up the slide, and that energy is ATP. So let's see a diagram of this. We have simple diffusion, high concentration to low concentration. Facilitated diffusion, we have molecules going through proteins from high to low, and then active transport, we have molecules going from a low concentration to a high concentration, but they need energy to do that. Now, osmosis is the movement of water across a membrane from a high concentration to a low concentration. So let's see how this works. The water here is in blue. The solute are these little particles here. They can't cross the membrane, but the water can in this case. And when water can moving equally in both directions, we have an equal concentration of solute on both sides. When there is more solute inside the cell, that means we have a higher concentration of water outside the cell. Again, water is going to move from high to low, so it here will go into the cell, and the cell will often swell or burst. The opposite scenario is where we have more solute on the outside of the cell and less solute on the inside of the cell. Relative to the outside, the water concentration is higher here, so this means water is going to go from high to low, which means it'll move out of the cell, and the cell could even shrink. So let's do one practice problem together. A cell is in a beaker with a solution with a higher salt concentration outside than inside the cell. What will happen to the cell? Well, you probably see a little hint here of water is going to leave, and that means the cell may shrink. All right, moving right along to photosynthesis. Remember, photosynthesis transforms light energy into chemical energy. It's how plants and other organisms harness the sun's energy in order to generate the glucose they need, which then can be used in processes like cellular respiration. So this is the general equation for photosynthesis. You might even see it written out like this, where we have sunlight, carbon dioxide, water, generating glucose, C6H12O6, and oxygen. Remember that photosynthesis occurs in the chloroplast organelle in plants. And also remember that ultimately, most of the energy for life on Earth comes from the sun. So photosynthesis is going to provide that connection between the sun's energy and the needs of the rest of living things. Now, it's not just plants that are going to use photosynthesis, but also algae, algae and bacteria, so other organisms can perform this process too. And the food that they're making is actually glucose, so that's important to remember. Here's again the equation in words if you want to pause to write it down. If we zoom into a plant and a leaf, onside the leaf here, those guard cells again, and it's a little blurry here, but we can see those chloroplasts in the micrograph here. And remember, chloroplasts contain chlorophyll, which is the pigment that absorbs those photons or the energy from the sun. Now, this is related to cellular respiration because the outputs of photosynthesis are going to be the direct inputs in cellular respiration. So remember, photosynthesis takes in sunlight energy, carbon dioxide, and water, and it is going to release glucose and oxygen, which we need for cellular respiration. So cellular respiration takes place in the mitochondria of eukaryotic organisms, and it's going to involve taking oxygen and glucose in, and then using those glucose molecules to generate ATP energy for the cell, as well as water and carbon dioxide as byproducts. Now, we need this process in every single one of our cells, so the oxygen that we inhale every day from the air is going to go to that cellular respiration process. And we know that the carbon dioxide that we exhale comes from cellular respiration. We also get the glucose for this process from the food that we eat. All eukaryotic organisms are going to perform cellular respiration. Here it is again in equation form. But remember that there's not just one type of respiration. We also have anaerobic respiration, or a type of respiration that occurs without the presence of oxygen. Now, a lot of organisms are going to use anaerobic respiration, even us in our muscle cells when we run low on oxygen. But remember that this process is a lot less efficient than cellular respiration, which involves oxygen. So we generate more ATP with cellular respiration that is aerobic than we do with respiration that is anaerobic. Now, one type of fermentation is going to be lactic acid fermentation, where glucose can be broken down to generate lactic acid as a byproduct along with ATP. There's also alcoholic fermentation, where glucose is broken down to generate carbon dioxide, ethanol, which is a form of alcohol, and ATP energy. Here's a quick chart comparing the different processes, so if you'd like to pause and write this down for your study materials, you can. And let's go back to the carbon cycle. So remember that carbon and other elements are constantly cycling through the environment through different processes, and two of those main processes that link carbon to the environment are photosynthesis and cellular respiration. So remember, plants and other organisms harness sunlight energy and take in carbon dioxide to generate glucose, and then that glucose is consumed by other organisms, and eventually the carbon in the glucose is exhaled through respiration. It can also be broken down in decomposition processes, and then when organic matter is burned, released as combustion, and then that carbon dioxide can go back into photosynthesis or absorbed by the ocean in some cases. Another cycle that's important is the nitrogen cycle. So you don't need to know all the steps of the nitrogen cycle, but nitrogen is very abundant in our environment, and we need nitrogen in the molecules in our cells. However, humans and many other animals and plants can't use nitrogen in the form that it's found in the air. So plants and animals rely on the nitrogen cycle to change nitrogen into a form they can use. And we notice that bacteria plays a really big role in the nitrogen cycle by converting nitrogen into different nitrogen products like ammonium and then nitrate, which can be taken up by roots of plants and then consumed by other animals. And then when organisms decompose, it's converted back into ammonium and nitrate again. We can also get nitrogen fixation into the soil through lightning strikes, which is pretty interesting, but just know that nitrogen is constantly moving throughout the environment just like carbon is. And so just like these elements are moving throughout living things, so is energy. When one organism consumes another, it consumes its molecules, which contain energy. And here we have a simple food chain, but remember that as we go up in trophic levels or energy levels of organisms consuming each other, we lose a lot of that energy. So the most energy is found at the bottom of a trophic pyramid. Most of the energy is lost at heat, and in a lot of ecosystems, we see the most organisms and the most biomass, the most actual mass of all the organisms in an area at the bottom levels of these pyramids. And then we see less primary consumers, and then secondary consumers, and then tertiary and quaternary consumers. So as we go up in trophic levels, a lot more energy is lost as heat to the environment. So ecosystems are very complex, and there's a lot of interactions between different organisms, and different factors like the amount of food, the amount of water, competition, diseases can affect how many organisms of a certain species can grow in an area. You might see a graph like this, which shows an organism growing and growing and growing until it kind of levels off at a certain point. And you should recognize this pattern as a graph for the carrying capacity of a population. So when it reaches the maximum level of population that an environment can sustain, that's called the carrying capacity, which we'll see in a lot of different circumstances. Now, ecosystems can undergo large-scale changes from things like forest fires to eutrophication, which is when we have excess nutrients introduced to a system and then one type of organism kind of overgrows and outcompetes others, and it's going to alter the environmental balance of an area. But other dramatic events like fire and flooding and earthquake can cause huge changes to an ecosystem too. Remember, for humans, we rely on a lot of Earth's resources, including renewable ones, which can be replaced by natural processes relatively quickly, and a non-renewable one, which really can't be replaced within human lifetimes. So like forests and trees can regrow relatively fast, and sunlight is renewable because we always have the sun shining on us. But other things like fossil fuels are limited and they can be used up by humans. And one of the main issues that we have in sharing these limited resources is human population. And we know that humans are a major source of environmental change. And so we have to keep an eye on how we interact with the environment and the biosphere to protect our resources and these processes. You might have heard of global warming, which is an increase in the average temperature of the biosphere. And we know it's mainly the result of humans burning fossil fuels, which adds gas to the atmosphere, causing it to hold more heat. And continued global warming can lead to rising sea levels and coastal flooding. But we can look after the health of our biosphere by conserving resources and cutting back on greenhouse gas emissions. Other disruptions that might influence an environment are things like the introduction of a native species, which can outcompete native organisms, introductions of diseases, which can wipe out native populations, or other human-caused habitat changes like urbanization or pollution, deforestation, or logging. So we talked about mitosis and meiosis. Let's go back to meiosis a little bit. Remember that meiosis is not going to produce identical daughter cells. They're going to produce what are called haploid cells, and in humans that's sperm or egg cells. And these haploid cells contain half the genetic information as the original parent cell. When fertilization happens, we combine two cells to have a complete fertilized zygote or egg, which has a complete set of chromosomes. Then that zygote or fertilized egg cell can undergo mitosis, grow and grow and grow until the cells are ready to differentiate into all the cells that are going to make up an organism. Now, you do not need to know all the phases of meiosis, but do know that we start with a duplicated set of chromosomes, just like we do in mitosis, and we're going to go through all the stages 2 times in order to end up with half the genetic information as the parent cell. So let's take a closer look here. We'll have homologous, or chromosomes that contain the same type of genetic information lining up together. Those homologous chromosomes will split apart in meiosis. And then when we get to the second round of PMAT in meiosis, we'll see the sister chromatids splitting apart just like we saw in mitosis. Now, different events in meiosis where the chromosomes don't split apart the way they should can lead to different chromosomal conditions, meaning that in those egg cells and after fertilization, we have a variation on the number of chromosomes that we would typically see in human cells. So you might have heard of something called trisomy 21 or Down syndrome. That's when we have 3 copies of the 21st chromosome instead of 2. And these extra or missing chromosomes can have varying effects on humans, from a variety of symptoms to having huge effects on the organism. These are what are called karyotypes, which is where we line up all of the chromosomes for a picture in different organisms. And so a typical biological female will have 2 X chromosomes, and a biological male will have an X and a Y chromosome. So there are different chromosomal conditions which can vary, and sometimes we see XXY or just a single X, XYY. There's a lot of different variations. So here's a look at trisomy 21. If you look at that 21st chromosome, we see 3 copies. Now, we know that we can have a lot of variation among humans, even among siblings in the same family. And some of that genetic variation comes from different ways that the chromosomes align and combine during meiosis. So one of the major things that happens is something called crossing over. When those homologous chromosomes line up during prophase 1 of meiosis, during that crossing over, the arms of some of the chromosomes are going to cross over each other, literally, and switch positions. So what we end up is kind of a nice combination of different traits from the 2 parents. We can also have these chromosomes lining up in different orders and combinations through independent assortment, which is going to allow us to have a different distribution of traits after the process of meiosis. Now, mutations can also occur, but some of the major sources for variation in different organisms are going to be from things like that independent assortment and crossing over. Now, remember that if there is a mutation, a change in the DNA sequence can result in a change in the protein, which can can lead to an altered protein function if it doesn't fold the way it normally does, which leads to a different phenotype or different physical characteristic in the organism. So this is Gregor Mendel. He's the father of genetics. So he determined that a single gene could have two different alleles or versions, such as a dominant and recessive version, and those different combinations could lead to expression of different traits in organisms. So if you want to pause and do this exercise real quick, you can identify which genotypes here, or combination of alleles, are either heterozygous or homozygous dominant or homozygous recessive. I'll put up the answer in just a minute here. There we go. And remember that the phenotype is the physical characteristic caused by the genotype of a different organism. So for here, if we know that yellow body color is dominant to blue, determine the phenotype of each fish. And at the bottom, we have gray being dominant to orange, so determine the phenotype of each owl. You can pause it if you want. Here are the answers. All right. Let's take a look at a Punnett square and how you would set that up. So in axolotls and other organisms, albinism is a trait that is recessive. So if two parents with pigments, they have at least one of the big A alleles, mate, what are our chances that the offspring will be albino or have this lack of pigment? So here we have this combination of two heterozygous parents. They both have pigment and they both have big A, little a. So we separate those genotypes on the Punnett square, one on each side, and then we're going to drop down the letters into each box of the Punnett square. And here are our results. So it looks like there's a 1 out of 4 chance, or 25% chance, that the offspring of this cross will have that albino phenotype. And there's a 3 out of 4 or 75% chance that it will not be an albino axolotl. Now, Punnett squares are fun, but it is important to note that most human traits are very complicated and caused by many different genes, meaning they're polygenic. There's also, so we know that a lot of characteristics in humans are on a spectrum and it's not one thing or the other. There are certain genetic conditions like cystic fibrosis and Huntington's that are caused by single alleles, but those are more rare. And more commonly, we have many complex genes interacting together. So one of those ways that genes don't work the same way that Mendel came up with are codominants. So that's when we have a combination of both traits in the offspring. Instead of having one or the other, we see both of the phenotypes appear. And here would be a cross. We use capital letters to represent codominant traits. Blood types are an example of codominance. When we see the AB blood type together, it's because an individual has a genotype of big IA, big IB. We use the I here to represent blood type because there's also type O blood, which is recessive to that capital I with the A or the B. So we can end up getting 4 possible phenotypes for blood type. You can be type A, type B, type O, or type AB, and those come from the different potential genotypic combinations. We're not going to get into the positive, negative, that's the RH factor here. That's for another biology class. But here would be how you would set up a cross for a type A homozygous individual and a type B homozygous individual. So we would put their genotypes on either side of the Punnett square, and it looks like if these two individuals crossed, we would have 100% chance of their offspring ending up to be type AB blood. Let's see it again with an individual that has a genotype for type that has type B blood, but they also carry a little I allele. Same thing over here. They have type A blood, but they carry that little I allele. And in this case, with this cross, we have a 25% of these two parents having kids with type O blood. And we noticed that they had the same phenotypes, so the same type A and type B in this situation, but because the genotypes are different, we get different results in the potential genotypic combinations for the offspring. All right, you also might see incomplete dominance, where instead of seeing both phenotypes represented, we see a blending of traits here. So the red and the white would combine to form pink. A lot of traits, too, can be polygenic or polygenic, and we see the distribution of these traits often on a bell curve. So if we see this kind of distribution in a particular trait, we can infer or we can guess that it is controlled by multiple genes. So in this case, we're seeing a trait for height, and we know that because there's this bell curve pattern on the graph, that it's a trait that's polygenic or controlled by multiple genes. Now, some traits are actually altered by environmental factors. Certain reptiles, their sex can be determined by the environmental temperature that they experience while they're developing. Another example is hydrangea flower color. Now, if they have different exposure to different pHs in the soil, they may end up being a different color in the garden. We can use DNA to determine how related or similar different species are, especially when we're trying to draw lines between evolutionary relationships. For example, we can take a look at some DNA samples or even protein samples or sequences of amino acids and proteins and see how similar they are to different species. So in this list, we have protein A from species 1, and in the table, we're showing that same protein in different species and how similar it is to that same protein in species 1. So if we look at these percentages, we can see that species 3 here is actually most closely related to species 1 because it has the greatest similarity in that protein A structure. Another great tool for determining relatedness or drawing connections in evolutionary relationships is gel electrophoresis. And that's when we take DNA samples, we cut them up with restriction enzymes, and we run them through a gel matrix with a current. So DNA is negatively charged. It'll be attracted towards the positive end of the gel, and it'll move down the gel in this direction as we run the current. And then we can see how similar the DNA is based on the separation patterns of the fragments. So for example, the mother here, we can see has a similar gene pattern with child A here, a similar gene pattern with child B here, but no similarities with child C. So this mother is probably not biologically related to child C based on their DNA banding patterns. We can also use this in forensic analysis. So if we have evidence at a crime scene, we can compare the evidence, the DNA evidence, to the DNA of the suspects accused of the crime. So if you want to pause it and try to figure out which suspect best matches the evidence at this crime scene, you can do that. So here's our evidence. And we note that suspect 3 is an exact match with our evidence here on our gel electrophoresis. And so we can figure out who committed the crime by matching that DNA to the crime scene. Now, gel electrophoresis is just one type of biotechnology technique. There are many others. And biotechnology broadly is when humans use organisms or parts of organisms and modify them to create products or tools for human use. Often, biotechnology is used to create drugs or other substances used in healthcare, genetic engineering, development of medical treatments, agriculture. Biotechnology is everywhere and it's interdisciplinary, and we have a lot of exciting things that are going on with biotechnology. So obviously there are ethical questions that come up with any new technologies. And so there's a lot of good questions that are out there from, using CRISPR to edit embryos, or bringing back the woolly mammoth, or doing advanced genetic screening on humans, or even sterilizing mosquitoes so that they won't go and grow their populations and bring new diseases to an area, and lab-grown meat. So all of these things are within the realm of biotechnology, and all of them bring up good ethical questions to discuss. All right, we're moving forward here to evolution, which broadly is the change in the genetic makeup of a population over time. Natural selection is a major mechanism of evolution, and a good term to know is evolutionary fitness, which is measured by reproductive success. So that is how likely an organism is to survive, reproduce, and pass on its genes. We know that environments are going to change and act as a selective mechanism on populations. And so let's take a look at what natural selection looks like up close. So this is a bacterial population, and here we have an antibiotic that a person takes because they have a bacterial infection, right? They have a lot of bacteria growing in their bodies. Some of these bacteria, the pink ones, they're going to be resistant to that antibiotic. So let's see what happens when the antibiotic comes. It's going to kill off most of the bacteria, but the ones that have the resistance are going to be able to grow and survive without the competition of the dead ones now. And so what we've done is we've changed the population, and the makeup of the population now is mostly antibiotic resistant. And so that is natural selection in just a few steps there. And so we see this with pesticide resistance, we see this with antibiotic resistance, or with any other trait over time where we see one trait that is more favorable in a particular environment, allowing those organisms to survive, reproduce, and pass on their genes more successfully to their offspring. Now, we see evidence for evolution in a lot of different areas, from the fossil record to geographic evidence to DNA and amino acid sequences, so this biochemical evidence to embryology and seeing similarities in how organisms develop. All of these things provide evidence that different species are related through common ancestry. Now, here's Darwin again. He was the guy who came up with this main model for natural selection. And his ideas behind natural selection are that species often are going to have more offspring than an environment can support. So there's a struggle for survival, competition for limited resources. And so the ones that have the traits best suited to their environment are going to be the ones that have the competitive advantage over other individuals in that species. And so When we go into different environments, we can see different variations of different traits in varying population and different success and survival rates based on those varying traits. Now, an adaptation is a genetic variation that is favored by selection and provides an organism with an advantage in a particular environment. But if the environment changes, that adaptation may not be as favorable. So it's all dependent on the environment that the organism is in. And sometimes we see things like similar adaptations in different populations. So here in the Arctic, we have natural selection driving the adaptation of white fur in a lot of different animals, like polar bears and foxes, because the individuals with white fur have a survival advantage because they can camouflage against the snow, which allows them to better hunt or prey or evade predators. So these organisms aren't all closely related, but they are all demonstrating the same trait because it helps them survive in their environment and it's beneficial over time. Now, again, like I said, environments can change, and sometimes, environments experience things like droughts, and in that cases, the organisms that are best suited to survive are going to be the ones that are drought resistant. And so we may see here in this environment certain species or individuals and populations dying off, and then other individuals that have variations of traits that are more suited to that dry environment and can withstand low water availability and high temperatures, being more likely to survive and pass on their traits. And so over many generations, we might see a complete change in this population over time. But within a single population, we may see variation. So here, this is just 4 different populations, and we can see that different ones have different variations based on the colors that are displayed. Now, if there's a change in that environment, let's say a disease that wipes out all the green individuals, the populations that are more diverse are actually going to have a better chance at surviving because some of them are more resistant to the disease. So it's always good to have diversity in a population because they're more resilient to changing environmental conditions. So let's wrap up with a review of the 3 domains of life. We have bacteria, archaea, and eukaryota. Eukaryota include all the eukaryotes like plants, animals, fungi, protists. So in these 3 domains, bacteria are familiar to most people and associated with disease, but there's a lot of bacterial species that do not cause disease and play beneficial roles in our environment and in biotechnology. And the soil is full of bacteria that perform essential functions in the biosphere. We have a whole bunch of bacteria living on us as part of our microbiome. Archaea is its own domain. They are prokaryotic, they're single-celled, and they are both best known as being extremophiles or organisms that live in really extremely harsh environments. And finally, eukaryotes, like I said, they are composed of eukaryotic organisms, protists, fungi, plants, animals, and they include you and me. So beyond domain, we have different levels of classification to group different organisms. You might have heard of acronyms for this, like King Philip came over for ginger snaps, but the scientific name of an organism actually comes from the categories for its genus and species group. And so scientific names are always italicized. The first word always has a capital letter. The second word has a lowercase letter. So these are some of the scientific names of common organisms. Gallus gallus is the scientific name of a chicken. Sus scrofa is the wild boar. Entamoeba histolytica is the amoeba. And Toxicodendron radicans is poison ivy. And every living thing that has been discovered is assigned a scientific name in this format. So in the eukaryotic domain, like I said, we have animals, we have plants, we have fungi, and we have protists. Protists are usually unicellular, but they are eukaryotic, so they're pretty complex. Fungi can be unicellular or multicellular. They have cell walls, and they're heterotrophic. Plants are multicellular. They have cell walls and organized tissues, and they're autotrophs. And then, of course, animals are multicellular, eukaryotic organisms. They don't have a cell wall, and animals like you and me are heterotrophs. So phylogeny is the study of evolutionary history and the relationships between different groups of organisms based on the idea that all organisms share a common ancestor. A phylogenetic tree or a cladogram is a visualization of evolutionary relatedness between species. The lines are not always drawn to scale. Sometimes they are, but oftentimes it's just displaying the relationships, not the length of time that different organisms branch off from each other. So we can also see traits that appear on cladograms or phylogenetic trees. So here we have each hash mark is a trait, and everything that came after it includes that trait. So, for example, ferns, pine trees, and flowering plants all have vascular tissue. Seeds developed next, so pine trees and flowering plants have seeds, and only flowering plants have flowers on this cladogram. We can look at common ancestors between different organisms based on where branching points meet. So here, if I put these letters on the tree, which letter in the tree corresponds to the most recent common ancestor of yeast and the worm? Well, if we look down here, that would be the letter D, because that's the branching point between those two organisms. All right, this is not everything you may have learned in biology, but hopefully it is a good start to your review. All right, you did it. You made it through the review for the NC Biology EOC. Thanks so much for watching. I do have other review materials and study tips on this channel, so be sure to give this video a like if you're enjoying this content, and don't forget to subscribe or share it with a friend if it's been helpful for you. Thanks so much for watching, and I'll see you later.